that encompasses both.
It's
Because you're saying a chain.
Awesome.
I know, but
It's nice.
Yeah.
Nice to learn. It's awesome.
Okay. What do you think I said? I didn't know what you were talking about. Just so.
I did make. Yeah, I made sure. I would think so.
So sorry.
I think for sure that's it.
It's crazy. How different like college experiences.
The rise.
Like,
Yeah.
everybody's like,
Yeah.
Overdue ones forever.
works hard, but like,
He probably goes to
It's
like
not
the
like
lab or
yeah, or he just lays in his bed and like,
Yeah, no, like-
yeah, but it's just so good.
It's the same because it is something.
The battles. Y'all just look at it.
I think actually.
We find that we found a different and then whatever that is, we found a different. It's so good.
Oh my god. Wait, are you looking?
No, I don't. We got interview.
Wait, because I don't know.
I like those.
Oh, yeah, I just.
So we are looking at protein, starting to look at protein structures today and the types of interactions that happen both between the backbone atoms and potentially side chains and to hold together a three-dimensional structure of a protein. So in this, we're going to start off by looking at secondary structures. So those are regions of a protein that are less than the whole protein. It would be smaller regions that have one common structure to them. The first one we'll look at is alpha helices, and these are held together by hydrogen bonding in an alpha helix in proteins. A turn occupies about 3.6 amino acids. So the reason why it's not an integral number is because the amino acids are held together by hydrogen bonding, and the hydrogen bonds are between the carbon and the oxygen that is part of the backbone of the polypeptide, and the nitrogen and the hydrogen below it. So this is the hydrogen bond donor, and this is the acceptor. And you get the hydrogen bond between those two. And so you would have every 3.6 residues, you have a hydrogen bond. And so you have many of those, not every 3.6, it's actually more frequently than that. But within a turn, every 3.6 will hold together those backbones. So you have many hydrogen bonds holding together the whole secondary structure and keeping it fairly stable. And in a structure like that, this is just showing the backbone. So you can see the structure of the backbone, but the side chains will project out from the alpha helix. So they're sort of coming out like this. And we'll see a top-down view of that. Okay, so depending on the composition of the polypeptide in the alpha helix, you can imagine that this structure will have a characteristic of being hydrophobic or hydrophilic, or we'll talk about amphipathic in a minute, but it will depend on the composition of the amino acids that make it up. So if you had an alpha helix that we called hydrophobic, what types of amino acids would it be composed of? Yeah.
Or non-polar.
Mm-hmm. Yeah, so non-polar amino acids. What are some examples? What are a few? Yeah. Alanine, phenylalanine, right? Tryptophan, yeah, things like that. Many of them. So that would make a hydrophobic alpha helix. Hydrophilic would be our polar amino acids, or acidic or basic amino acids, and so on. Okay. The second secondary structure that we'll look at are beta strands. And beta strands are a little more extended in structure. So the backbone has an extended conformation, but it's sort of pleated. And so sometimes you hear the term beta pleated sheet. This is a strand. And if you have another strand next to it, it can form a sheet. And they are held together also with hydrogen bonds between those same atoms in the backbone, the carbon and the oxygen, and the nitrogen and the hydrogen. This structure, this image is a little misleading because it looks like two proteins. But really what's going on is that we have a region of the polypeptide that's in a beta sheet. And then we might have something like a loop out here, and then the protein continues back in this direction. So it loops back on itself, and it forms these hydrogen bonds between them. And so it's a single protein. It is interacting with itself. And this is showing the directionality of the chain on each side. This is the N-terminal end and the C-terminal end. And if it loops back around, it's N to C. And that is called an anti-parallel beta sheet because it's N to C and N to C. Okay. And nice hydrogen bonding between all of those atoms. We can also have a structure that is a parallel arrangement of beta strands. In that case, it's just the directionality is the same. N to C, and then how does this form? Oh, maybe the rest of the polypeptide loops out and comes back in the same direction next door. So that is called parallel beta sheet. You get hydrogen bonding between the same atoms there. Okay, it's just looking at the directionality. Okay, questions on either of those structures? All right, another secondary structure is called polypeptide or polyproline helix, which probably makes the most sense. And that is because the structure tends to form when you have multiples of prolines in the composition of the polypeptide. Prolines have this ringed structure where the side group ends up coming back and forming a covalent bond with a backbone. And that structure ends up forming a bend in the backbone of the amino acid backbone side chain or backbone structure here. You have multiples, and they call it a kinked structure. So if you have multiple of those in a chain here, it ends up turning the backbone a bit. And it turns it in it's a little hard to see here, but a left-handed way. This turns to the left. And what's different from this secondary structure from others we've seen is that doesn't have to be held together with hydrogen. Okay, it just forms that structure because of the kinked nature of the backbone, because of the prolines. When we see a polyproline helix like this, the structure ends up typically also having a lot of glycines interspersed here. What do glycines look like? What is their side chain? It's just hydrogen, right? And so, right, there are two hydrogens, but the side chain itself is just a single one. There's always the other hydrogen on the alpha carbon. And we intersperse glycines there, or biologically that tends to happen because glycines are so small. it allows this kinked structure to form. You don't have steric problems with a large side chain, being in competition for space with these ringed structures. Okay, so this is a look at both the alpha helix and beta sheets in a different orientation. So you can see the backbone and then the side chains. So we were looking at an alpha helix sort of up and down structure. If it's laying on its side, here you can see the side chains projecting out from it. And this is a top-down view. You can see the side chains projecting all around the alpha helix there. A beta sheet, we were looking at it face on, where we could see the hydrogen bonds. And if it's flipped on its side, you can see that the side chains project above and below the plane where the hydrogen bonding is occurring. So you can see that there are almost faces to these two structures. There are sides to them. And so because of that, these individual structures could potentially be amphipathic. What does amphipathic mean? Yeah.
Hydrophobic.
Yeah, so it has both hydrophobic and hydrophilic characteristics. So how would an alpha helix be amphipathic? If you had the amino acid side chains that are all on one side of the alpha helix, let's say they're all hydrophilic, and the other side is hydrophobic, if that continued throughout the structure, you'd have a sidedness to it. You'd have one side that was hydrophobic and one side that was hydrophilic. So that alpha helix could be at an interface, like between a lipid bilayer and the aqueous cytoplasm, something like that. The beta sheet can also do that. In this case, it's pretty simple to create an amphipathic beta sheet because each alternating amino acid side chain projects on either the above or below the structure. So you can have hydrophilic, hydrophobic, hydrophilic, hydrophobic, and so on. And that would create a sidedness to that structure as well. Alpha helices, you would maybe have 1, 2, and then 3, 4 would have a different characteristic, and then 5, 6. Because remember, the rotation is about every 4 amino acids. So there are about 2 on one side and then 2 on another, and another 2 coming back to create a rotation. So either of those can be formed in that way, depending on the composition of the molecule. Okay, so we'll be seeing, and if you ever look at scientific papers that study protein structure, often they show the structure in sort of a cartoon form, which leaves off a lot of the atoms so that you can see the basic structures. So these are beta strands. Each one of these is a beta strand. And that's what the arrows, the thick arrows mean. The arrows will indicate the directionality of that part. So this is the N-terminal end, and that's the C-terminal end. This isn't showing any of the connecting regions of the polypeptide. So we're leaving that out. This one does show the looping between the 2 beta strands. So you can see the other parts of the protein there connecting. And the directionality of the arrows, because it's indicating N to C terminal regions, are these, let's say these 3 beta strands, are those parallel or anti-parallel conformation? Those are parallel. These would be anti-parallel. Okay, so that also indicates that directionality. Sometimes if you have many of these together, they end up twisting on themselves. And if you have enough of them, they can form what they call a beta barrel. It ends up sort of looping back on itself and forming a ring structure. And sometimes these ring structures can potentially be embedded in a lipid bilayer and serve as a transporter for things going into and out of the cell or into and out of an organelle. And so sometimes these beta barrels are amphipathic. So the outside, the R groups that are facing out towards a lipid bilayer would all be hydrophobic. And the R groups that are facing into the middle of the beta barrel might be hydrophilic. And that would allow molecules to go through that are hydrophilic and pass through a lipid bilayer in that way. It's a channel system. Okay. So we'll get back into that idea, but that can be the usefulness of something that has the amphipathic characteristic. All right, questions about those? Yes.
We'll also have like the X.
Yes, yeah, thanks for pointing that out. Yes, this structure here indicates alpha helix. So yeah, this is a beta barrel, and then would have one alpha helix there, and then a bunch of somewhat unstructured loops connecting the beta strands. Yeah, thanks. Anything else? Yeah.
So both are hydrophobic and hydrophilic, like the outside of that transporter, for example, is hydrophobic.
Yes.
So like that would be able to kind of pass through a phospholipid bilayer.
Yeah, so it would be something like this, right? You could think of this as being the outside of those beta strands. Those would all be, the outside of the whole thing would be hydrophobic, and the inside could potentially all be hydrophilic. Yeah, so that's how that would work. And things in that structure, there's proteins called porins in kidney cells that allow like tons of water to come through. So that's useful for that. But there are other proteins that have that structure that are used for that as well. Yeah. Right. Okay, so how do we form these folded structures? We talked last time about how the peptide bond doesn't rotate, right? That rotation is restricted. Why is that?
Because of like the partial double bond quality?
Yep, because it has a partial double bond quality. It doesn't rotate, but there are in the backbone of a peptide, the phi angles, which are between the N and the alpha carbon, and between the alpha carbon and the carbonyl carbon, those do rotate. So those are called the phi and the psi angles. Those can rotate, and that allows all these proteins to fold up into the various structures that we're seeing here. So theoretically, those can rotate a full 360 degrees. Whether they do that or not is going to depend on all of the atoms that are connected onto those chains and whether they're interacting with each other in space or trying to. So you can have steric clashes between those if they're in angles of rotation that cause that problem. So a scientist named Ramachandran set out to map what are the allowable angles of the phi and psi bonds, and which ones do we actually see in a protein chain that are allowable with regard to steric problems. So I'll explain what's going on in this graph. Each axis is the full 360-degree angles of rotation of phi and psi angles, and each point in here would be a coordinate of two of those. So these are all the possible allowable angles. And what he asked was developed a computer program that would model the combinations and used a polyalanine chain. So just a chain of alanines, and asked what are the allowable angles there. What he found was only in the white regions are the rotations actually allowed, because in all the other potential angles, there were steric problems. There was really a small subset of allowable angles when you did a model in this way. And what he also found when he mapped onto those regions, where were these secondary structures that are most typically seen, those fall within those angles of rotation. So we have our right-handed alpha helix, our parallel and anti-parallel beta strands, our polyproline helix. There are a couple other helices. We sometimes see one as a left-handed alpha helix instead of right, turning the other way. And then this is another alpha helix called a 310 helix. It just has a slightly tighter turn to it. So instead of 3.6 amino acids, it's 3. Those common secondary structures all map to those regions that are predicted to be allowed. So then what he wanted to ask was, okay, these are theoretically allowable angles. What has actually been documented on protein structures that have been solved? So looking at alanine residues and actual real proteins from X-ray crystallography data. And those are shown in gray. And those correspond very highly to a lot of these secondary structures, and even quite often in a tighter region than the white outline. There are a few other things we see outside these regions, which might be random turns, random coils, but most of them map, of the proteins that have been solved, most of them map within these regions. So that all aligns. And just for reference, not something you need to memorize, but these are the ranges that are required to adopt for each of these secondary structures. So a beta strand has to have phi and psi angles in these ranges, and that's how you can adopt those structures. Okay. So of the theoretical allowable angles of phi and psi, real proteins really adopt a very small subset of those actual angles of rotation. Okay, so let's look at an example of a protein and examine it in a little more detail. Proteins fall into sort of two really broad classes. One are fibrous proteins and the other globular. Globular tend to look like blobs. They're a little more spherical and blobby, and fibrous are long and extended like fibers. These are a few common fibrous proteins. We're going to look at collagen today. And we can see outlined in, highlighted in red here, the most common amino acids in each of these fibrous proteins by quantity. So if we look at collagen, do you have any predictions, just straight off the bat, of types of secondary structure that this might adopt? Does it have a lot of, yeah.
Maybe like a polyproline.
Yeah, so I think I would agree. Maybe a polyproline helix that has a ton of prolines relative to these other fibrous proteins. It also has a lot of glycines, which are required to facilitate the kinked structure of the polyproline helix. So that is true. So let's look at collagen and what it looks like. Collagen is the most plentiful protein in vertebrates. It basically forms the structure of all of our connective tissue. And we look at the diagram here, each one of these strands is an individual polypeptide. So the green is one polypeptide, the yellow is one, and then this white blue thing. So that structure is that left-handed polyproline cage structure. And it has sort of a repeating sequence of amino acids. There's glycines, there's prolines, and there's what's called hydroxyprolines, which we have not heard about yet. And that's a modification to a proline. We'll talk about how that happens. Okay, so lots of repeats, which gives it that left-handed curved structure. Now, in building a collagen molecule, 3 collagen polypeptides come together and interact in this sort of twisted structure here, sort of turns on itself in a right-handed way. So this is a triple helix called a tropocollagen. And that is held together, we'll look at this in a minute, by hydrogen bonding, highly facilitated by the hydroxyproline group. Okay, we'll come back to that. So that is a tropocollagen, a 3 helix structure. And then we get another level of structure to this. If we look at this diagram, this is a cutaway view of tropocollagen here. So this would be a long tropocollagen molecule. You get multiples of those interacting with each other to form something like this, quite thick. And this is called a collagen fibril. So these are held together with the tropocollagen, are held together into a fibril by cross-linking between lysines. And we see quite a few lysines here too. Okay, so we're mainly going to focus on this level of structure though. That has hydrogen bonding between a lot of the amide protons and the carbonyl carbons of neighboring amino acids between each of the polypeptides, and also the hydroxyproline. So that is going to stabilize that 3 helix structure. All right, so this is our hydroxyproline. You can see that's potentially a hydrogen bond donor or an acceptor on proline. Okay, so where did we get this? What's hydroxyproline from? That group, this hydroxyl group, is usually added after the proline is translated. So the protein is made in the cell and the hydroxyl group gets added. I'm going to show you the chemical reaction to do that. You don't need to memorize that, but there are a couple points to take away here. So this is the enzyme that adds on the hydroxyl group to a proline. And the point of this reaction here is that in order for this to occur, it requires the presence of ascorbic acid or vitamin C. Right? This is going to be a cofactor in the reaction of this hydroxylation. So if you do not have vitamin C, you cannot get formation of hydroxyproline, and that potentially weakens the hydrogen bonding structure between this tropocollagen molecule. And if you have extreme deficit in vitamin C, it causes a disease called scurvy. Has anyone heard of that before? Yes. What context have we heard of this? Pirate, of course. Scurvy pirates. So the reason why that is, and that's not specific to pirates, that occurred when people were at sea for months at a time and they had no source of vitamin C. They had food, but nothing had vitamin C in it. And so over time, they didn't get formation of hydroxyproline. It weakened the tropocollagen and weakened essentially all of their connective tissue. And that causes all manner of problems, as you might imagine. Hemorrhaging beneath the skin, loss of teeth, joint weakness, bleeding. You start to have mental problems, hallucinations. It's very painful, and there's no, at a certain point, the damage is done. So even if you have vitamin C, there's a certain amount of repair that can't be regained. So very serious. Once it was figured out that this was alleviated by making vegetables or citrus, which, you know, survive a long time without going bad, they just brought things like that on the ships. So that is the result of a lack of vitamin C there, and at the level of the protein structure, what's going on. Okay, questions on collagen structure and the formation of that? Yeah.
You said that extreme vitamin C deficiency leads to something which weakens collagen fibers, so hydroxy.
The lack of hydroxyproline. So because you can't add this hydroxyl group, you're reducing the amount of hydrogen bonding that can occur between the polypeptides at that level of structure. Mm-hmm. Okay. Now a look at tertiary structure of proteins. Tertiary structure is really the whole three-dimensional structure of polypeptide. In something like a fibrous protein, especially like, so if we look at, sorry, about going back. If we look at the tertiary structure of one of these polypeptides, it doesn't change a lot. It's just a polyproline helix over and over and over again. So the secondary to tertiary structure doesn't change a lot. In globular proteins, that is a little different in that we might have regions that are alpha helices and then other parts that are beta sheets. And then the whole structure together has a whole three-dimensional look to it. And that's what we call the tertiary structure. That is typically stabilized by a lot of the non-covalent bonds we've been talking about. But at that level, we can also get one type of covalent bond between side chains, and that is called a disulfide bond. And that happens between the end groups of cysteines. If the cysteines lose their protons that are at the end of the sulfurs, you can get two sulfurs coming together to form a covalent bond. And that can stabilize a tertiary structure as well. People tend to study protein structure by X-ray crystallography or NMR. We also have pretty amazing computer programs at this stage like AlphaFold that can predict tertiary structure just based on a protein sequence, which is amazing. That was not possible for, up to the past, 5 years when that was developed. So that's an amazing accomplishment. And then the tertiary structure is basically going to determine a protein's function, what it does. Yeah.
So you said the cysteine bonds are part of the tertiary structure?
Yes.
Oh.
Yeah, the bonds between the end side chains there. Mm-hmm. Okay, so this is just a look at tertiary structures of some proteins. Now you should know what these individual secondary structures indicate. And then the overall structure, its composition. So this one, for example, has a beta barrel and 3 alpha helices, and the entire structure is described here, including the loops that connect those secondary structural regions. One aspect of proteins that, especially enzymes, is that an enzyme might be catalyzing a reaction. That's its job and its function. But even within that protein, there are specific regions called domains here that have particular functions to that enzyme. So A here is the catalytic domain. That's the part that catalyzes the reaction. C is the regulatory domain. That tells the enzyme whether to turn on or off. So a domain, just by definition, means that there's a particular function associated with that region of the protein. And that might also be associated with a particular structure, but it might not. But sometimes domains are associated with a particular structural form. This is a look through a beta barrel. And so these structures are quite stable. They're stabilized by covalent bonds potentially between the tertiary structure, but lots of non-covalent interactions will stabilize these structures. Yeah.
Would tertiary structures be considered domains of quaternary structures?
Typically not. A domain is more related to a function. Quaternary structure is the presence of multiple individual polypeptides in a final structure. So yeah, I wouldn't say that a tertiary structure is a domain because that's not necessarily functional. Yeah, that's mostly related to the function. But yeah, good functions. Okay, and then we might also see in some of the turns that occur in a polypeptide, a tight turn like this in the backbone is called a hairpin turn. Very tight. There are multiple types. You don't need to memorize those, but you can note that they are often held together with a little hydrogen bonding there too to hold that stably. Okay, last part I want to talk about here is the entire folded structure and a bit about the energetics of this. So when a protein is folding, there is an energetic quantity associated with that. And because proteins tend to fold, the folding tends to have a negative delta G. There's a drive towards folding the protein. One of the main initial driving forces of protein folding is the hydrophobic effect. And what is that? What is the hydrophobic effect?
The cages or no?
Mhm.
So it's just like say there's like a non-polar substance like a lipid inside, and so it's going to form a cage to sort of protect water hydrophilic from the non.
Yeah. Right. So the hydrophobic molecules or the water is trying to avoid interacting with hydrophobic molecules. So it forms these caged structures, ordered water around hydrophobic groups. The same thing can happen around hydrophobic side chains. Okay. So we know that lipids, let's say in water, tend to glom together into droplets. Sort of the same idea happens with side chains of amino acids in a watery environment. The hydrophobic side chains try to hide from water. And so they tend to fold up into the center of a protein if the protein's in an aqueous environment. So if you look at a sequence of a protein, this is a long protein, are the green amino acids hydrophobic or hydrophilic? One letter codes here.
Phobic.
Those are hydrophobic. So the greens are hydrophobic and the reds are hydrophilic. And if we look at those side chains in the context of a protein like this, you can see that the hydrophobic groups are folding into the center of that protein. And the hydrophilic side chains are facing out towards water. And that's because they don't mind interacting with water. Okay. So you might imagine also, if you were in a lipid environment, the opposite would occur. the hydrophilic side chains would fold up into the middle and the hydrophobic chains would be out. So it depends on the environment, the folding, the tendency to fold in a certain way. Okay, so the folding is driven by the hydrophobic effect and the increase in entropy in water.
You have a combination of both, right? Separated just to show like the different.
Oh, yes. Yeah, they're separate. This is all a single protein here. Yeah, and this is just showing the directionality of the side chains. So when a structure is folded, the entropy of water can increase. And we also have favorable interactions. We have the hydrogen bonding, we have, side chains interacting in a favorable way. So that's also contributing towards the favorability of protein folding. But it is an ordered structure. So the protein is losing entropy when it folds. Okay. The water might gain entropy, the protein loses entropy. And so you can imagine that much like nucleic acids, things can unfold at higher temperatures, right? So all of a sudden, if you raise up the temperature, the directionality of protein folding might change. So we will get into that. Okay, I don't want to take up all the time. Let's have a little time for the problem set, and I can answer questions.